Welcome back to Vaccine Design course. Our next step is tertiary structure prediction of the antigenic protein. Okay, so for that, the next step that we'll be doing is homology modeling. Now, you need to take the protein which is antigenic and has many epitopes for homology modeling. Okay, so we have done all these steps to determine or to shortlist the protein which is antigenic and has many epitopes. Now, to talk about homology modeling, it obtains the 3D structure of a target protein based on the similarity between the template and target sequences. Now, homology modeling, it's the process of predicting a structure from the sequence. So, and that should be comparable with the experimental results. So, this particular protein structure is determined by its amino acid sequence, right? And these structures can be obtained from X-ray crystallography, NMR spectroscopy, or from theoretical methods using real-time experiments or by computation method, which is called as homology modeling. So in this, a 3D structure is constructed by aligning a target protein sequence with the known template structure. So you have a target and a template. Template is what is known and target is your protein sequence that you have already determined by all these steps. Okay, so now this protein sequence, they can be obtained from NCBI or UniProt or PDB. So as you can see in the slide, the figure on the right hand side, you have a template, unknown structure, then you will do the sequence alignment and then you will get the model. So this is done by homology modeling. Now, the quality of the structure depends upon the extent of similarity between the target sequence and the template. So the highest similarity should be taken. Now, it cannot be used to predict structures which have less than 30% similarity. But in rare cases, less than 20% can be selected because there may not be many sequences related to some proteins. For example, when you take the HPV68, we take the less than 20% because there are not many sequences related to that particular protein. Okay. Now, coming back to the homology modeling, it's a multi-step process which includes sequence alignment, structural modifications, database searches, energy minimization, and structure evaluation to generate a structure. Let's see the basic steps involved. First, protein target or query sequence retrieval has to be done. Then template sequence or the template structure retrieval has to be done. So we have the target as well as the template sequence that is retrieved. Now you need to align both the sequence. Then there is model generation and validation. So here you can see in this figure, it says the step-by-step model where you need to identify, align, then sequence, loop building and evaluate the model. Okay. So now to generate these models in homology modeling, there are two ways. One is the automated method where we only need to paste the target or query sequence and rest of the steps are done by the automated server itself to generate the models. The second mode is the alignment mode where we perform all the steps and only the fourth step is allowed on the server. Okay, but the other steps we perform by ourselves. So the fourth step is again model generation that is done by the server. Okay. So now how to choose between the automated and the alignment mode depends upon the template. So if you don't find your desired template, then prefer automated. On the other hand, if you have a desirable template, as per your choice, you have to choose the alignment model. So to perform this homology modeling, both online and offline platforms are available. In this lecture, let's do the automated modeling. Okay. So we are going to do the automated mode. So first, the server that we'll be looking for is Swiss Model. So go to Swiss Model, create your account and wait for it. So this is your Swiss Model homepage. You need to log in if you have your login details or you need to create an account. Okay. So that you can get your results instantly in your account itself. That's why. Okay. After creating the account, you need to press Start Modeling. Then you will get this site or you can also click modeling my workspace. Okay. Then what you need to do is paste your target protein sequence. Okay. So format must be FASTA, which we already have. Control C and then Control V. Then after doing it, if you want to enter your project title, you very well can enter. And then what you need to do is click on Build Model. When you click that, you'll see that the models will appear here when it's ready, and now the auto model is running. So when you get the results, you'll see all the models that you have obtained. Okay, so now the results are ready. As you can see, the automated modeling did with 24 templates, and the models that you got 3 models, right? One, the second one, and the third one. So you have 3 models. Now to download this, what you need to do is you need to go to project data. Then you have something called as archive. Okay, so when you do this, your zip file will be generated. Next, let's open it. So we have opened it, and then what you need to do is you need to unzip it, and then you can see something called as Swiss model. So take the Swiss model. So we haven't named our project, so take the untitled project name. Then you have models. Open the model, and then you can see the 3 models, 1, 2 and 3. So model 1, you can see the image here if you want to see in PNG format. Yeah, so this is the image of the first model. See, it's similar. Okay, the PNG format, and you could see the other formats as well. Okay, so this is how you need to download it. So you can take one model. So let's just take the first model, okay, where you can see the stability and the alignment is shown. Okay, so model template alignment, let me just take, yeah, so these are the stability data. You can see the coverage information, the sequence identity, which says 19.05%. it's only the percentage identity is just 19.05. okay, so this was the model 3. Now when you go down, model 2 is 18.85, and here you can see the model 1, the first model, which is 100% identity, and you can see the coverage also. So we'll, if you're taking this protein structure, and let's say this protein structure is antigenic in nature, non-allergic, and has many epitopes, you can take the model 1 because the sequence identity is 100%, right? When you see, you can see the model template alignment as well, which is beautifully shown here, right? Yeah. Now when you go to the summary page, you will get about the template, the sequence identity, and the description of it. Okay, and also the other information of your template. So the first one, the template was this, and now there is something called as GMQE. Okay, so here when you go, it's you can see it's global model quality estimate. Okay, there is Q means Z-score analysis and others. Okay, so this is how you create your model using automated modeling, right? Now let's go to another server, which is called as Fiery2 protein fold recognition server, and see how the automated and modeling is done in this particular software. So search for Fiery2 server in Google and click on the first result. Okay, or you can also use this particular link as well. Okay, so this is your home page where you need to provide your mail address. Then if you want to give your job description, you can give it, and then what you need to do is paste the sequence and make everything default if you want, or the modeling mode if you want only normal. If you want intensive, you can go for that. That depends on your aim, and then you can press for Fiery search. Okay, so let's just copy the amino acid sequence and paste it here, give the mail ID, and press the Fiery search button. So here are the results. You can actually download the zip of all the results like how we did for the Swiss model. Then you can see the different models, and this shows the top model which they have designed based on template, this particular template. Okay, so now you can see the PDB header, chain, title, entry, and next you can see what is called as the confidence and coverage. So confidence, it's 100%. It means the stability. So how much stable your protein structure is, and coverage is again 100%. So you have 100% stability and 100% alignment. And now this is your 3D structure. You can view it in JSmall, and for other options you can see the FAQs, and then you can see the various model dimensions that they have written. And now if you want to see the secondary structure and disorder prediction, you can again go for this. You can again have your PDF report by saving it here, and then the domain analysis is shown here, and the detailed template information is also shown here. Yeah, these are the template information and various models. So the first one was the top model which was seen, right? Yeah, so here when you see the domain analysis, it shows the templates that are being used, okay, in the domain analysis. So there are 15 templates that the model has used, the Fiery2 server has used. And now, as I've mentioned, you can see the secondary structure, disorder prediction, and now you can also see the structure of each template when you hover the cursor over it, right? Yeah, right. Okay, so just hover your cursor over each template, you can see the structures of it, right? Now about the secondary structure and disorder prediction, here you can see the sequence secondary structure, that is the alpha and beta sheets. Now the secondary structure confidence, SS confidence, which determines the stability. Next you have the disorder and disorder confidence. So confidence key from highest to lowest is indicated here, right? From red to blue is indicated. So red means it's high and blue is low. Next what you can see is again domain analysis, which we've already covered. Then you can see the structure of each templates that are being used, and yeah, and the template information. Then you have the different models, right? So this is how you have a better interpretation in Fiery2 than you see in Swiss model, right? Yeah, so maybe you can go forward, but you can use the two servers and then find out the differences. Okay, so that's about the automated modeling. Okay, so we studied two servers where automated modeling can be done: Swiss model and Fiery2 protein fold recognition server. So in the next lecture we'll be learning about the manual modeling or the alignment modeling. Thank you.